So welcome to CTSI, Translational Tuesdays. We'll wait a minute or two while people begin to log on. My name is Dr. Ralph Sacco. I'm director of the Miami Clinical Translational Science Institute. And we had instituted these special webinars during COVID. And we have them now approximately once a month. So this is really our August webinar, even though it is September 1st. So this is a webinar. Most of us do a lot of Zooms. So this one's a little different in that we have the chat feature to let us know if there are problems. And we have the Q&A for you to type in questions that we'll handle towards the end of the webinar. It is a webinar and we are recording it. So it is recorded for others to take a look at that aren't here today with us. And this is an ongoing series. We have these all listed now on the CTSI website under our COVID resources area, where you can see a lot of special projects that are ongoing at the University of Miami Miller School of Medicine, including some of these webinars that have been recorded there. We also have a short poll in the end that we ask you to take so we get some feedback on the program. If I can have the next slide. So this is a very timely Translational Tuesday webinar, and it's really all about convalescent plasma for the treatment of COVID-19. It's critical because, as you'll hear, we've been selected to participate in a very highly important NIH-funded, NCATS-funded trial that Dr. J will tell us about. And it's the importance of our role in this and other upcoming randomized clinical trials on convalescent plasma. So we have really three great experts, local PIs involved with these convalescent plasma trials. Dr. J will open with a discussion on the importance of randomized controlled trials to validate convalescent plasma for the treatment of COVID-19. He'll nicely show that, the data isn't as clear as we think. And the need for a randomized trial to really, truly show efficacy for this theoretically important treatment. And then Dr. Shweta Anjan will follow with an overview of upcoming convalescent plasma trials launching at UHealth on the outpatient side, which will be even more critical. And she's the PI on an important study that she'll tell you about that I think is going to maybe even either be approved today or tomorrow. And then Dr. Yanyan Wu, who's in the Department of Pathology, will touch on what the FDA's emergency use authorization means for these trials. And she'll talk about the EAP data that we actually contributed to and how that EAP data helped inform the FDA in their emergency use authorization. But I hope you'll see that there are still some holes. So Dr. J is Professor of Medicine, Infectious Disease Research Unit, Medical Director of the Clinical Translational Research Site, and one of our Associate Directors in the Miami CTSI. Dr. Anjan is an Assistant Professor of Clinical Medicine, Infectious Disease Division, and PI of one of the trials that we'll be talking about in the Department of Medicine. And Dr. Yanyan Wu is Medical Director of the Transfusion Medicine at UHealth and Medical Director of our Transfusion Services at Jackson Health System. And the transfusion services are critical for these convalescent plasma trials, and she's in the Department of Pathology. So without further ado, I'm going to turn it over to Dr. J to get us started.
Thank you, Ralph and Sheila and Daru and Raquel and all my co-presenters. So I feel like one of Henry VIII's wives, I have very short time left, so about 20 minutes. So I'll try to accomplish this during that time, and to be followed by my co-presenters. As Ralph said, that we are going to talk about convalescent plasma, but this morning you can see on the New York Times what a bad job we have done. As a nation, we have dropped the ball completely, and you can see the difference between different countries, and compared to Germany, South Korea, and Japan, we have done very poorly. So the question is, what went wrong, and how can we correct some other changes? So today's topic is convalescent plasma. So the issue is, where does where does convalescent plasma fit in this pandemic, and how we can use this to improve the survival and the mortality and morbidity of people with COVID-19. So the history of convalescent plasma goes a long time. This is a publication from JAMA in 1937, where they were using for pneumonia. So there is some evidence that there can be some help with convalescent plasma using in diseases. For example, this slide came from FDA in 2006. So it came when the avian influenza pandemic started. They found that about 60% of the people who were getting were dying. And it was a desperate situation, and they were trying to use convalescent plasma, and they were preparing in anticipation that in case it came to US, that FDA was prepared. Now we can say that, COVID has come. There were 25 million people infected, with many others dying, and so it's something that we are thinking that how does convalescent plasma fit in? So historically, the FDA argued that in a pandemic where vaccines, effective antiviral drugs, respirators, antibiotics may not be available, which will lead to high mortality, maybe the convalescent plasma can be made locally. The evidence exists that sometimes it could save life. So that's what the argument the FDA was making. So was there general evidence for efficacy? There are no actual clinical trials. Since 1892 until the antibiotic era, serum was used in diphtheria, pneumococcus, meningococcus, measles, mumps, the list goes on. Even and then they use specific immunoglobulins for HPV, CMV, rabies, RSV. So you can see that either the immunoglobulin or plasma has been used before, but there has not been any trials. For example, Argentinian hemorrhagic fever, there was a 90% of people died. They tried it, and there was some success. So the question was, is there any preclinical data? So there is some clinical data, not randomized controlled trials with all the bias fit involved, but is there any preclinical data? So there have been publications in 2006 when the avian flu was coming in. There was a mouse model they created pneumonia in that model, and they used equine hyperimmune globulin and monoclonal antibodies, and there was some benefit. So of course, fortunately we are not mice, nor are we non-human primates. We are humans. So all these studies does not actually mean that it will work on humans. And that, of course, there are the other studies. So is there anything that suggests that it works in flu? Of course, again, this comes from the FDA in 2006. The meta-analysis of Spanish flu, there were 8 evaluable studies, and some of them showed some benefit. But still, there are no well-controlled, randomized controlled trials. Of course, the Russians have some data, but, in Russia, things get approved quite easily. So this was again from Luke from 2006, looking at different studies to see if there's any response. And there seems to be at least 2 studies showing some favors treatments, about 20%, but these were not very well controlled studies. So absolute 25% less mortality in patients treated with convalescent plasma in the 1918 flu pandemic. So these are all historical data. We can't really be 100% sure how valid this data is. So basically, if you have an infectious disease, what the sequence of events are, and basically you identify the pathogen, which the Chinese did, and then you do the genome sequencing with the Chinese did and shared with us. Then the convalescent plasma is used usually. Then the vaccine comes and then goes to prevention. So this is the sequence of events that normally do. So my talk today is basically on convalescent plasma. So this was using at randomized controlled trial. So this slide is actually quite important because you can see that there are really important things that we have to consider. So this slide shows that the initial 3 studies are on COVID-19, the balance are on MERS and COVID-1. So those are a different era. The important thing to remember is that the way you use convalescent plasma will show how well they work. The timing of the administration is really important. Earlier you give, better the response. And the volume you transfuse, the more you give, better the response, because you don't know how much the antibody titer is. And of course, the antibody titer, you have to have adequate antibody titer for it to be effective. So unless you control for all 3, and unless you control the condition of the patient, these apples and oranges cannot be compared. So you can't just cherry pick studies that work and ignore the studies that didn't work, because that way you could seriously get into bias situation. So this is the way basically the donors are collected. It has to be make sure that your donors recover from disease, and you have to give adequate time till the antibodies are generated. You must make sure that the antibody levels are there, and you have to have resources to develop these. And then the antibody titer should be more than 1 to 320. I think Dr. Wu will talk about it. I'm not going to touch upon that. And the standardized amount of plasma should be collected. You must find out the the specific product to be produced, whether fresh frozen plasma or fresh plasma or lyophilized plasma. This is blood bank expertise. It's not mine, so I'm going to skip that. So it involves a lot of technical details. So this slide is actually quite important. It just shows that the best time to collect the plasma is about 46 days. So the whole thing went crazy. The FDA commissioner issued emergency authorization, which Dr. Wu will talk later, basically said that based on totality of scientific evidence available to the FDA, it is reasonable to believe that COVID-19 may be effective. But the point is, nobody else thought that that was correct. In fact, if you look at if you read it through, then they say, given that the clinical evidence supposing EUA was not obtained from prospectively well-controlled trials, additional RCT must be continued. So basically, he's saying one thing in the first page and completely opposite in the second page. So that was a huge problem for us. So we as clinicians and researchers, we are caught between the devil and the deep blue sea. So when we looked at the Cochrane library, as you know, the Cochrane library basically it goes to systematic review of different keywords, and they took 20 studies with 5,400 participants, and about 5,211 received convalescent plasma. There were 50 were randomized. So the bottom line is this. They say overall risk of bias is very high in all studies. Uncertain whether convalescent plasma has any effect at all on all-cause mortality in at hospital discharge. Uncertain whether convalescent plasma prolongs time to death, and uncertain whether convalescent plasma has any effect on improvement of clinical symptoms at 7 days. So basically, Cochrane Library, looking at all the data, says there's nothing here. There may be signal, but there is nothing here. The CNN comes back and says the FDA makes this decision desire to appease the president rather than a serious consideration of science. And the real consequence is that this will affect how we can enroll patients. So, and there are a lot of scientific leaders like, from the Pittsburgh, and Rob Kelly from FDA, former Commissioner Eric Topol from Scripps, and then the Soumya Swaminathan from WHO, and Mark Macklin and Margaret Hamburg and Scott Gottlieb. They're all previous FDA commissioners said this is not right what FDA did. So when I have this thing, I discussed with Roy Weiss, and he quite rightly said, why don't you talk to Ken Goodman? So Ken sent me this, ethics paper, which analyzed this issue. So first thing is that he said, the first principle of EU allocation should be to minimize harm. So we should minimize harm. The convalescent plasma must be for the people who are really sick. That's correct. It's critically important to do a randomized control study. Assuming the convalescent plasma works and giving it freely does not minimize harm where there is scientific equipoise. So for us to say that it's unethical not to give convalescent plasma, which is available by order, is actually not very ethical, because we know that there is no data to support it, just because legally we are told that, but ethically, the real ethics is that morally, we know that there's not enough evidence. So the ethicists say EU may be used in places where there is no RCT available. But if the RCT available, unless the patient refuses, you should do this, because you need to prevent group harm by doing a randomized control trial. So ladies and gentlemen, now I come to the studies that Ralph quite rightly, I'm thankful to Ralph for bringing me into this. And these are the list of NIH funded studies. I'm not going to go one by one, but you can get the picture, like, all the who's who in the NIH and the research community is involved in this. So there must be something right in this group, because, they won't be in this position unless they are in that. So then we come to our study. So this is the NYU, Einstein, Montefiore, Yale convalescent studies. Now we added Ralph Sacco, who is the PI for this study. I'm just a piano player. I'm the clinic. I run the clinical trial bit along with my colleague Anjan and Dr. Wu, but the PI is Ralph Sacco. So fundamentally, we are trying to answer the question that the ethicist and the scientific community and the previous FDA commissioners and Sanjay Gupta and all this, and Tony Fauci and everybody is saying. So we need to do a randomized control trial. We are looking at the people who are sick with respiratory symptoms, hospitalized less than 72 hours, who are on oxygen supplementation. And we are going to give high quality plasma with high antibody titers, which means that, as you remember the slide that I showed, that the best time to collect was about 46 days. In the last line of that, it shows that the antibodies against the receptor domain. So if you collect good samples, they have a lot of antibodies against the receptor domain, which really works. And of course, it's placebo controlled. And the primary endpoint is this. The secondary endpoint is base score per the WHO ordinal scale. And we are supposed to enroll 180 subjects. And this is my slide I give slide credit to Daru and Sheila and Ralph for making this slide. And basically, it is the same thing that I said. So, and now you get the picture. So in summary, the history in a pandemic, the vaccines, antiviral drugs, respirators, all these are needed, but convalescent plasma can be made locally and potentially can save life. But the FDA goes and throws a wrench into this whole thing by saying it's approved. But the whole world knows that it is approved, but not with data, so data has to be created. Cochrane says that we are uncertain whether convalescent plasma has any effect on all-cause mortality or prolonged time to death, or effect on improvement of clinical symptoms at 7 days. Therefore, the only solution to this confusing, dystopic situation is to do a reverse randomized controlled trial, since EUA was not obtained from prospective, well-controlled randomized trials, and additional trials are needed. With that, I will stop, and I'll let my colleague Shweta take over. Thank you. Now I have to stop. I have to stop sharing, right?
It's at the top, Dr. J.
Oh, okay. Stop sharing. Okay. Oh, okay. Okay. No. I have to. Did I stop? Or? Okay, I got it. Yeah. Sorry.
There you are.
Go ahead, Dr. Anjan.
Good afternoon. I'm Shweta, one of the infectious disease physicians here at University of Miami and Jackson Memorial Hospital. I work with Dr. J, Dr. Wu, and the whole team here. So thank you, Dr. J, for that great update on everything related to convalescent plasma and the randomized controlled trials. I think the one update that I will be giving or talking about is the outpatient clinical trials, which is new for this pandemic. As so far in the past, all the trials that have been done for convalescent plasma have been inpatient and used to treat severely ill or critically ill patients. What we know so far from the data presented by Dr. J and what Dr. Wu is going to highlight later would be that most of the data supports safety of the convalescent plasma transfusions. There is some mortality benefit, especially when you give it early after diagnosis. But what we need is definitive evidence. COVID-19 outpatient management. So far, we have none. Outpatients would have no access to convalescent plasma for infection prevention or early treatment, except to the ongoing randomized controlled trials. Society would not return to normal function in the absence of outpatient vaccines or therapy. The presence of outpatient management or outpatient trials would definitely decrease the burden on the emergency room and hospital admission rates. Participants can be a part of the solution to rapidly finding out whether the antibodies delivered through convalescent plasma work or not. And honestly, military and civilian populations are depending on us to find the solution so that they can complete their missions and return to functional society. So we have actually been selected through CTSI for the 2 outpatient trials sponsored by Johns Hopkins and Department of Defense to be a site. It is a multi-site study. There are 26 sites already enrolled and a couple more coming on. There are 2 trials. One is to stem coronavirus, almost like a prophylaxis study. It is a randomized controlled double-blind study comparing efficacy and safety of human coronavirus plasma versus a control, which would be plasma obtained prior to 2020 or prior to December 2019, when the COVID-19 pandemic started. The PI for the trial is a Johns Hopkins professor, Dr. Shmuel Shoham. The second trial we are also a site in is for early treatment, which is convalescent plasma to limit coronavirus-associated complications. It is also a randomized controlled study for outpatients with symptomatic COVID-19. And the PI is David Sullivan. So a quick run through of what these involve. So the purpose of the first trial, or the post-exposure prophylaxis trial, it is to find out if convalescent plasma can prevent COVID-19 exposed people from becoming infected. Their goal is to try and enroll 500 patients, adults, basically household contacts, healthcare workers, anyone exposed to a known COVID-19 positive patient in the past 120 hours can be enrolled. It does not exclude immunocompromised patients. They have to have a negative molecular test for COVID-19 within the 24 hours preceding the infusion. The exclusion criteria is anyone who received a blood product in the past 120 days, symptoms or lab evidence of COVID-19 infection at the time of screening, and a previous allergic reaction to blood products. These patients would be followed up for up to 90 days. Obviously, as I stated before, it's a randomized controlled trial. You either receive 1 unit of COVID convalescent plasma with known antibody titers of greater than 1 to 320, or normal plasma that was collected prior to December 2019. The timetable so far was that prelim results would be available mid-October and final results in November or December 2020, but that might change. The endpoints the trial is hoping to reach are to evaluate the efficacy of the treatment at day 28 following the high titer plasma versus control in exposed subjects. And determine the incidence of development of COVID-19 disease after receiving convalescent plasma. Also determine the incidence of disease severity between the two groups if they develop COVID infection. Compare antibody titers between the two groups. So the other study, CSSC 004, this is for COVID-19 positive patients. The purpose is to decrease risk of hospitalization or death, duration of symptoms, and duration of viral shedding in COVID-19. The goal is to enroll 600 patients between all the sites. And we enroll symptomatic early outpatient disease. And the goal is to enroll them within 8 days of diagnosis. Pregnant women are not excluded. The plasma source is the New York Blood Center for both the trials, or if the local site can procure their own plasma, that is also allowed. The endpoints are hospitalization or death by day 28, and then the secondary endpoints are viral clearance, resolution of symptoms, antibody levels at day 90, and assess if there are any secondary infections among the household contacts. They also hope to compare serum antibody titers and rates and duration of SARS-CoV-2 RNA positivity between the two groups. We hope to have some prelim results or by December 20, but again, with everything going on, the timeline might shift. So the current status of the trials are, as of last week, there were 81 transfusions that occurred among all the sites. 25 for the infection prevention or prophylaxis trial, and 56 for the early treatment trial. As you can see, there have been issues recruiting, not just at our site, which we haven't started yet, but at all the other sites in the country. So a quick updates from our team. This is our study team. It's not just the people listed, actually a lot more people. There's a lot of support from the CTR staff, CTSI, IRB. And we will be participating in both the trials. We have 2 clinic sites, the UHealth CTRS clinic and Jackson Health System ACC East Clinic, which is where the COVID-19 patients would be seen for early treatment. And at our CTRS site, it would be all the negative patients for prophylaxis. The estimated number of patients to enroll would be 50. Our current status is that all the agreements have been signed and we're pending SIRB approval, which is expected this week. Hopefully tomorrow, but let's see. So if you do have any questions, feel free to reach out to the study email or directly to me. I think it is important that we participate and enroll patients into the outpatient trial, because this way we would be able to keep our patients outside of the hospital and manage them early. At the same time, it is also important that we prove convalescent plasma actually works and not follow anecdotal evidence. I am done with my presentation and I will hand over the talk to Dr. Wu.
The correct screen. I am gonna start. So thank you so much, Dr. Angie, and also Dr. Jay for the nice overview. And so I'm mostly gonna talk about the implication of the emergency use authorization from the FDA. So up to the point of EUA approval, and so we basically have 3 pathways to access convalescent plasma in the United States. The first one is expanded access program run through the Mayo Clinic, which is actually a lot of the information that we will go over later on, which is a significant a part of the recommendation from the FDA is actually based on expanded access program. Then there's also a significant number of patients that are actually going through the single patient emergency IND process. FDA has those information. They're also analyzing that information as well. And then we also have the ongoing clinical trials. So the basis for the FDA to make the EUA approval are basically based on the following information. One is the history of convalescent plasma, Dr. Jay presented, and then evidence of a preclinical safety and efficacy data in animal studies, and then published study on the safety and efficacy of CCP, which is abbreviation for COVID-19 convalescent plasma. And then the last important one is the Mayo Clinic-driven EAP process. So we mostly will present briefly, because the time is very limited, of the last two categories. So the first one for the EOA approval base is basically ask the question, is COVID-19 convalescent plasma safe? So in short, the information is largely coming from the Mayo Clinic Expanded Access Program. So it's a very large cohort of patients. They had two publications on this one. One is the early one, basically on the 5,000 patient, and then the most recent one based on the 20,000 patient. So the rate of a transfusion reaction was less than 1%, and then thrombotic events was less than 1%. The cardiac events was roughly about 3%. But in short, basically based on the data and expert review, as well as the general knowledge from the field, the adverse events level is basically comparable to regular plasma infusion. And a lot of people anticipating or thought there might be the potential so-called ADE effect. None of them were truly observed, although that might be something we continue to watch. And so again, the information regarding the safety is in general safe. Based on the information so far in the United States, there are more than 70 to 100,000 units already being infused, and Florida contributed at least 10,000 of them. So then the next question is really what's the basis for the EOA approval? I'm just going to present sort of some simple summary of the information and the clinical data that so far has been published. The first two, I think it's important to go over a little bit in detail, is the two very important RCT, and that actually gave us a lot of information that's also helped us to design better clinical trials moving forward. The first one is this JAMA article is talking about RCT and was done in China early February to April. Unfortunately, the initial design was 200 patients, but they have to end it earlier when they enrolled 103 patients due to the control of endemic. So basically, in a way, it's a good thing, but they had to stop the trial earlier. So the key thing about this trial is very well-balanced trial in terms of the control arm, which is just the standard care, which do not have a placebo, and then versus the treatment arm. Treatment arm is patient receive convalescent plasma with a titer on 1 to 640 and above, which we'll go over a little bit of what that titer means later. But in short, there are summary of the outcome information based on the time to clinical improvement for patients with COVID-19 within 28-day period. The figure, the 2A is really all patient combined. You see a little bit separation from the convalescent plasma versus the control. And then you have the middle one, which is the patient with severe COVID-19. It's pretty much the patient, they're not being intubated yet, they're not in total sepsis, etc. Versus the life-threatening disease, which is the patient's already intubated or have a severe sepsis and on ECMO, etc., etc. So in general, you can see clearly in the life-threatening disease, there's really no separation of the control arm versus the convalescent plasma arm. But in the severe disease arm, you do see a separation. The p-value was 0.03. However, the study was considered as negative because when the group interaction was applied to the statistical analysis, and that was negative. So the other thing is, has been sort of buried in most of the message is regarding the antiviral activity that has been observed, because this information was not very obvious in the abstract, and the data was actually in the supplement. So it's a very interesting to note. Doesn't matter what group of the patient, either all the patient combined, a severe versus life-threatening disease, when you compare the control group versus the treatment arm. So within 3 days, all of them, you observe a significant higher rate of the viral conversion from positive at baseline and then to negative. So this is basically indicating convalescent plasma does have possibly the antiviral activity. And this one is definitely clinically statistically significant data. So because of the result was negative, but the JAMA the editors felt this was still important message here. So they had this editorial written, a randomized trial of convalescent plasma for COVID-19. Despite it was negative results, they did want to make a note there's potential hopeful signals. So the key findings about the study was early termination RCT, non-blinded, non-placebo controlled. One of the main things that's notable was their convalescent plasma was given pretty late. The median duration from the symptom onset was 30 days. But they were giving their high titer basic unit. The titer of 1 to 640 was basically based on anti-S receptor binding domain, which is roughly equivalent to 1 to 80 in terms of neutralization titer. So I have noted about the antiviral activity was observed, and there was a hopeful signal in the severe COVID patient, but was really negative finding in the severe or the critical life-threatening patients. The second important study was the RCT. That wasn't really published yet. It was mostly based on the preprint information. The study was from the Netherlands. They also terminated their study earlier. But the basis for their termination was quite interesting, was also quite controversial. They terminated their studies because they found a majority of their patients, even though it's quite early, only 10 days from the symptom onset, they reported basically those patients already had positive antibodies. And their level, when they compared their patient cohort to the donor cohort, they say the antibody level was equivalent. This is a quite interesting finding, which is somewhat different from the Chinese study. However, the Chinese study, they actually had a exclusion criteria. If the patient had a titer more than 1 to 640, they basically did not include those patients. So this is the finding that they found. They thought it was important enough to let the information out to the other community. So this is very controversial because we actually at this point do not know. The ICE receptor binding domain antibody is the only one that's contributing to the effect of convalescent plasma or not. And their data actually shows this is sort of human data, but not in human, sort of in vitro data, that maybe other antibodies are as important. So that's not known at this point. So again, the key finding is early termination. Their result was negative finding, but then the study was terminated earlier, and they also noted high titer in the donors already at baseline. So the next one is the 3 CT control trials. I won't go into much detail, just other than showing those are real study. One in the Middle East, and like 21 patients versus 28 in a control. And then the other one is the Middle East study, one more in China. All of them are small number, the study with mixed signal, and all have significant study limitations. And then there were also a number of cohort retrospective matched control studies. Similarly, mixed signal, mostly positive, against significant study limitations. And then the next one is a very important one for the FDA to make the decisions, basically based on the EAP data. You can see very large participation from the United States with a large number of patients enrolled. At this point, at least more than 700,000 already. A lot of people heard from the news, try to get the information, where was that 35% come from, etc. Somewhat is this figure, not entirely accurate information, but this figure summarizes the key finding from the study. Number one, if you gave the convalescent plasma earlier, within 72 hours in non-intubated patient, if you gave them high titer plasma, they seem to do better in terms of the 7-day mortality, but it's also some data on the 30-day mortality seems to be relatively better. And then, and this is other sort of significant finding of the study, is the high titer versus low titer, and the high titer patients seems to do better than the low titer patient. This is a similar study. One of the byproduct, I guess, of the EAP study design is actually giving us that sort of the continuous value of the continuous variables, so which actually help for the future study design for sure, is basically because it's a mixed bag of everything in this very large cohort study. So you're able to find where, for example, what is the levels antibody that actually should be the cutoff. And also what are the days that's you should start giving the convalescent plasma. So in a way, it's a very informative, very useful study, but doesn't necessarily providing all the information we need, but that's contributed to our knowledge and help us to design better study. So this one is basically the information with the 7-day adjusted mortality, basically based on the ortho IgG group, and you can see the separation of the information basically based on the titer level. So this is really the basis for the FDA to form the EOA recommendation. So the importance of this finding is that they are, if you gave the plasma early, convalescent plasma earlier, you may realizing some of the mortality sort of relatively maybe lower risk in terms of mortality, but then importantly, the gradient for mortality was seen in relation with IgG antibody levels in transfused plasma. So again, it's a very informative, very important study. Will help with future study design, but that doesn't answer all the questions we need to. So now I'm just gonna go over quickly what is in the elements of the EUA. So the convalescent plasma based on EUA has to be collected by FDA registered blood collections facility. Will have to from the individual, they contain the entire, basically the several SARS-CoV-2 antibodies. And then of course, the donor need to meet all the donor requirements. And then because of the EUA data that's informed them on antibody level, all they can do right now is basically say, if you follow the ortho, basically the assay that the Mayo Clinic used, if the signal ratio cutoff is 12 or above, you can label them as a high titer. And then if you have a different assay, basically you have to require amendment from the EUA to the EUA from the FDA. And in addition, if you have a plasma, they're not high titer, not meeting the criteria as high titer, you could still use them, but you have to label them as COVID-19 convalescent plasma of low titer. So those units you could use, and the healthcare provider could decide how to use them based on the risk and benefit. So FDA probably will continue to monitor and change your rules, which they have been. So the proposed indication is very wide. They basically say anybody hospitalized with COVID, you can treat. Okay, and then they also emphasize basically some recommendation of dose is basically standard care for transfusion, gave 1 unit, and then watch for the volume overload, very generic. So at this point, for the pathway for us to getting convalescent plasma for the patient, expanded access is already ended, no longer exists. Single patient emergency IND, very limited. If you have something that's not covered with the current very wideness of the EUA, you can ask for it, but pretty much non-existing. Then mostly it's the clinical trial, as Dr. J. Dr. Anjin discussed. And then you also have this EUA, I call them a transition phase. Because right now we're not EUA yet, because blood centers are not testing the titer most places. Even they're testing the titer, they don't have the titer label ready. So the FDA basically granted a 90-day extension for this EUA transition period. So they're not EUA. We have to tell the patient this is what's called investigational COVID-19 convalescent plasma. And the products are not labeled with a titer. And basically for 90 days, you can continue with this. So in summary, convalescent plasma with adequate antibody titer can be collected from patients, a recovered patient. They appear to be safe. They're associated with antiviral activity. If convalescent plasma given earlier may be associated with a clinical improvement. Then we probably have a lot more questions in terms of products. What is the antiviral activity? Which antibody at what level? And then is it other than antiviral activity, do we have any other rules contributed by the product? And which patient should we give at what time, any other factors? So basically many, many unknowns. This is the reason why from the FDA, when they issued the EUA, they emphasize giving that the clinical evidence supporting this EUA was not obtained from prospective, well-controlled, randomized clinical trials. Additional RCT are needed. They also emphasize convalescent plasma should not be considered as a new standard care for the treatment of patients with COVID-19. Again, they encourage all providers to enroll patients in those ongoing clinical trials. Thank you very much.
So great. Thank you very much, Dr. J, Dr. Anjan, and Dr. Wu. I'm going to first correct Dr. J. I may be listed only as the contact or administrative PI because this came through the CTSI in terms of the funding, but Dr. J and Dr. Wu are the real and principal investigators of this in-hospital trial, and Dr. Anjan is the principal investigator for the outpatient trial. I think before we get started, and there are some questions, Jay, can you just put up what makes this trial unique? It's in hospital, it's early, it's using high titer plasma, and can you talk a little bit about the outcomes just in terms of inclusion criteria, just briefly? We don't hear you, Jay.
Okay. I'll show you my screen inclusion criteria. So basically, this study is looking at people who are more than 18 years of age, who are hospitalized, and they have to have respiratory symptoms. It's funny because now we are seeing a lot of people with GI symptoms, but in this study, we developed it for people with some respiratory symptoms, with some hypoxia on oxygen. And within 72 days, so as Dr. Anjan and Dr. Wu said that earlier you treat, they're better off. And of course, people who cannot consent, we can use a proxy, and it has happened with us. The exclusion criteria we have people who have received full immunoglobulin within the last 30 days, contraindication for a transfusion, people are already on ventilation, mechanical ventilation or ECMO, they are excluded, but we still accept people on other forms of non-mechanical, non-invasive ventilation. Then we have to be careful of the congestive heart failure and as what you said that unlikely to succeed. So these are the things that that we use as an inclusion-exclusion criteria. This is FDA regulated study, so it's as IND, so we have to make sure that we are responsible for all the regulatory requirements.
Great, thank you. So this trial started at Einstein. Lizanne Porofsky, whose name you saw on one of the slides, is the principal investigator. She's one of the chiefs of infectious disease at Einstein. NYU got involved. They got to about 180, 190 patients. There is a DSMB that monitors this study, and they haven't called it to stop early, and instead the call was made to get other CTSIs where COVID was increasing to get involved. So they reached out to Miami, Houston, and actually Yale have now joined in this trial. And as you can see, it is a unique trial. We have some questions that I want to try to get to, and I'll distribute to some of you. So first, it is a randomized trial, and these are double-blinded. And believe me, it's not easy, and some of our CTSA colleagues have how we go to double-blind the convalescent plasma as best as possible. I don't know who wants to address that in terms of maybe how the blinding is done.
I can attend that. So we are quite used to blinding these things because we have done stem cell studies in ISKI where we blinded. We wrap the whole thing in different colored tubing, different color bags, and then we cover the tubing. But sometimes it's not that easy because if you put saline, when nurse tries to infuse it, they have to scan it. So there are, we have done some creative methods of blinding. At least all of us are blinded, maybe one or two are blinded.
And just to be clear, Dr. Anjan, just your study, they get normal plasma versus the convalescent plasma. So both are getting plasma. In the hospitalized study, it's placebo. I mean, it's saline, correctly. It has a covered bag and tubing and colors to blind it. But you are getting another plasma. So the blinding is a little easier in your study, right?
Correct, exactly. So essentially the investigational product would look the same. The only difference is the presence of specific antibodies or not.
What if something is in plasma that is protective? I mean, maybe something else that's in plasma. I mean, one would never know. And that's why, at least in the hospitalized study, it's truly a placebo in terms of saline. But we have some other questions. Somebody asked about how many plasma donation treatments can be extracted from one donor. This sounds like a good question for you, Dr. Wu.
Yeah, thank you so much. So the plasma collection is done by apheresis. It's somewhat volume dependent. So up to 4, sometimes it could be collected from one donor.
And remember, we're using the New York Blood Center for this. Plasma is all being done there, and it is all being quality controlled in terms of titer. Somebody else wrote, how do you know that the plasma works in the trial? Well, I think that's the point. The trial, I think, will hopefully show that it is working. It is of good titer, and you saw the EAP data, but we need a trial to really show efficacy. We still aren't sure that it is truly efficacious until you have really the right grade A evidence from a randomized trial. Jay, you would agree, right?
Absolutely, absolutely. Because, there are so many confounders like Dr. Shweta and Dr. Wu said, because, I mean, depending on the titer, depending on how quickly you give, depending on how much you give, it all varies.
Right. And somebody else talked about the difference that over time with mortality falling, could that have an effect? The other additional measures. Now remember, in a randomized trial, both groups are getting state-of-the-art care, dexamethasone, and now remdesivir. So hopefully, you'll see that balance out. But there always could be confounding if it's in a smaller sample. But we're hoping as we get larger numbers that we will help control for some confounding. There is a hope that if you get larger numbers, you can stratify among young versus older, comorbidities versus not comorbidities, earlier versus a little later. These subgroup analyses that would be important to really inform us about the use of convalescent plasma. There was another good one in here, I think, about shelf life of plasma. Dr. Wu, how about you on answering that one?
Thank you. So if the plasma collected was kept frozen, they're good for 1 year. And if we have to thaw them and they gave it to a patient for some reason that got delayed, we could keep them basically for 5 days with sort of extension. We changed them to thawed plasma. The plasma collected and then never frozen, they might be good for 26 days. So there are many different variations, but the longest one currently, if we don't change that, that'll be up to 1 year.
And they were sending us plasma to store for us going forward, right? We have a supply that will last 26 days or more, or how did they send it to us?
When they send it to us from New York, they're kept frozen. So they basically have 1 year for us to use. It depends on the day they were collected.
Great. There was another question here about, dose response. We talked about that. I think you nicely showed that in the EAP data. We'll have that data as well. We're collecting blood samples that we'll send up north for a central processing, and there'll be other ways for us to collect the titers. But did you want to also add to that, Dr. Wu?
Sure. So again, the information regarding titer can be quite confusing because there are many different ways to do the titer. There's an antibody assay, there is a true viral neutralization titer, there's also pseudo-viral titer. So they all correlate somehow. So the titer for the study will be done basically by all those methodologies, both antibody assay as well as the neutralizations for us to be able to find that right level. And then also which particular antigen that we're targeting is another question. So for this study, all of those will be well studied. So thank you. The implication of the study will also have some implication basically for, what will vaccine work, what antibody should be monitored. So that's a very important information to be studied. Thank you.
And Dr. Wu, I guess because people out there and some people who want to be donors, is there a wait time between donor donations?
So good question. So right now the FDA sets sort of a rules and it changes all the time. But in general, if you recovered fully, as long as you have a positive results. But actually, in fact, we actually have many donors that have donated basically without knowing they actually had a COVID. And so practically, we have many donors currently that were knowing that they just have titers. So that for that information, the best way to do is go to the One Blood website. They have all the information on what you needed.
There's a great question that I think we're all grappling with, which deals with the challenges of conducting a trial when we have this expanded use authorization. And I think we've all hit on that, all of us who were speaking today. In the best of circumstances, the CUA does confound it a bit, but I think as Dr. Wu told us, some of the EUA, which was rushed a bit, isn't even ready for prime time in terms of implementation. So, and even in the FDA statement, if you really read the FDA statement, it really says to prioritize randomized clinical trials. But Jay, do you want to touch on that again? And maybe Dr. Wu, I think it's a critical question.
Yeah, I think, I mean, looking at the ethics paper, even the ethicists find that pushing plasma without proper studies would actually do more group harm than less harm. So although people think that it's unethical to put somebody on a placebo-controlled study when they can get it freely, but not knowing what the other people on a non-randomized study are getting, it could be, may not be the best for the patients because the data is unclear. I'll let Dr. Wu clarify more.
Yeah, and Dr. Wu, clarify again, because you talked about, the process of implementing the EUA is not really even started yet. So there's a question here. Basically, we can give plasma to any hospitalized COVID patient under EUA. Is that true, and if so, how?
It's a very interesting. So when we started the convalescent plasma program in this country, nobody has assay set up. So basically, FDA was asking, as long as the patient recovered, we can use them. The figure that Dr. showed earlier in terms of timing, that was actually a study done in China that shows by what time the donor could have antibody. And those antibody, it could be quite variable, depends on the donor you're selecting. So we practically, at this point, we do not know the titers from the convalescent plasma we're using. For a blood center to be able to implement in testing a label, it's a very controlled process. It would take some time, set up the assay, to train the staff, to basically have all the quality control. So that's why FDA gave 90 days for everybody to get ready. So we're practically in the pre-EUA phase. We're not under EUA yet. Basically, we'll give out the plasma without knowing the titer.
Right. So it's one thing to have the television and the media and the FDA, right, we have an EUA, and it's another to say it started. So there is this window, and that's why it's even more important for us to put patients in the randomized trials and get the most effective answer.
Yeah, and I forgot to mention, so during this transitional time, FDA basically tell us we have to tell the patient, this is not a convalescent plasma under EUA. We have to, on the consent form, says this is investigational COVID-19 convalescent plasma, every single consent.
Right. There's a lot we need to learn here, for sure. And I think our three speakers today did a great job of putting this into perspective. You know, there's a lot of hype, and there are some politics involved here as well, as one can understand. But we're trying to do everything we can, the best we can, using science to guide our evidence-based treatments. And so I think it's great that we are part of this. I think we've heard from the experts today, Dr. Jay, Dr. Anjan, and Dr. Wu. Thank you so much for being part of this webinar and for being the principal investigators of these really important trials. Before we conclude, I think there is a little webinar question here. Please rate today's seminar on a scale of 1, 4 to 5, excellent. And how do you plan to attend, or do you plan to attend a future CTSI Translational Tuesday COVID-19 webinar? We hope to have more of these, and hopefully soon to be able to tell you about results of some of these studies and some other studies that are out there, maybe even the vaccine trials that we're part of. So I want to thank again our speakers and our principal investigators. They're doing a tremendous job. We're also going to get this up in Jackson and hopefully maybe even at Jackson South and North to increase the number of patients that we put into these important trials. Thank you for tuning in to our CTSI Translational Tuesdays.
Thank you, Ross.
Bye.
Yeah.